Hey guys, and welcome back. We are going to do the topic 11.4 review for gametogenesis and fertilization and all that stuff. It is a lot, so I'm going to go through it as quickly, but hopefully as in-depth as possible. So let's go ahead and get started. Gametogenesis. A gamete is just a sex cell in your body. Genesis means to make or the origin of, and so gametogenesis describes the various metabolic processes that create your sperm or egg, if you happen to be a male or female. Spermatogenesis is when we produce sperm, and oogenesis is when we produce eggs. So, three important things to note about all gametogenesis is that it occurs in the gonads. That would be the testes for you males, or the balls, and it would be in the ovarian tubes within females. And we're going to look at that in a little bit. So we're going to start with multiple mitotic divisions, no matter what, of precursor cells. Precursor cells are going to be diploid cells that are specially differentiated to be part of your germ line. So if you guys remember, it is very deadly and dangerous to have uncontrolled cell growth in most of your cell lines, and it's very deadly and dangerous to have a ton of genetic diversity in most of your cell lines, because most of your body cells, if they're skin cells, should be pretty close to the other skin cells in your body. Otherwise, your skin is going to look like a patchwork quilt, and it's going to function differently from one area of your body to the next, and that is not ideal. So there is a specialized germ line or reproductive line of diploid cells, and they are going to divide rapidly via mitosis, and they are going to grow into hair gametes. So that is what we call precursor cells. We are then going to have the meiosis or sexual division of these precursors into those haploid genetically distinct daughter cells, and the differentiation of these daughter cells into what we know as our gametes. Let's go ahead and get started. Spermatogenesis takes place in what we call seminiferous tubules of the testes. If you look at the testes via a cross-section dissection, you guys can see that they have a bunch of little veins and tubes going through them, and those are going to be where this takes place. It begins at puberty, go figure, when we have a basal layer of cells called the germline epithelium of the tubules, essentially just the inside cells of the tubes, begin to divide. They are precursor cells. They'll divide via mitosis into spermatogonia, which will grow larger into spermatocytes. So to get a good visual image of that, here is the seminiferous tubules. Here is the inner or basal membrane of those tubules, just the cells on the inside. We see the spermatogonium are the product of division, and then we have spermatocytes are going to grow out of those. Notice that we are still in a diploid or two in or two of each chromosome phase. Okay, so we haven't gotten to that haploid stage yet. All right, we're then going to have those big, large grown spermatocytes are going to divide via meiosis. They have to be kind of large because they have to split all of that material into 4 different cells. And they're going to divide into 4 haploid spermatids, which will differentiate or turn into spermatozoa through the activation of various transcription factors and genes. So spermatogonia, spermatocytes, spermatids, spermatozoa. And we can see that here after the spermatocytes divide, they go ahead and turn into the spermatids, which go ahead and turn into our functional spermatozoa. I told you guys to picture it in this flow diagram like this, if it helps you, where you can see the stark divide between the 2N and the N, the diploid and the haploid. And then I said it's kind of like Pokemon, where they evolve into different stages until they become spermatozoa. Woo! But if that's disturbing and you don't like associating sperm with Pokemon, which I really can't blame you for, I'm totally fine if you just don't use that analogy. Okay, so make sure you know the parts of the sperm. Here we have the archosome, which is going to be part of the cap. It's going to be the part that's going to dissolve later on when the sperm actually fuses with the egg, and it's going to activate all those transcription factors and everything. We have the haploid nucleus, which is going to be the genetic information the sperm is delivering. The centriole, which is going to help the sperm, once the centriole and the nucleus are injected into the egg, the centriole is going to help to perform to spin the spindle fibers that are going to allow for the division of the developing zygote. And then here we have, of course, our mitochondria, which are going to provide power for this tail, which are in a 9+2 arrangement. That just means that they are in 9 plus 2. It's like Okay. 9 plus 2 arrangement, I'll go ahead and show you 9 plus 2 arrangement in flagella. It looks like this, okay? So we have 9 sets of 2 is what I was trying to say. And it's just basically describing the protein fibers that make up the tail or flagella of the sperm. It allows for that rapid circular movement that they do to move the sperm through the fluid. Okay, so let's talk about oogenesis. So that was it for spermatogenesis. You don't need to know much about it, but you do need to know about oogenesis, and you do need to be able to compare the two processes. So spermatogenesis takes way less time. It's a lot simpler than oogenesis. Oogenesis is going to take place in the ovary and the oviducts. We'll talk about different names for those in a minute. But it's going to actually happen before you even turn into an embryo and a child. So it happens in fetal development. 40,000 of these primordial cells called oogonia are going to be created by mitosis. That's our primary mitotic division that happened in sperm in the basal membrane, but here with these primordial cells. These cells are going to grow into our primary oocyte. So just like we had on the last stage, the original cells produced by spermatogenesis grew into a larger form. Same thing happens here. Primary oocytes. And then our oocytes begin meiosis just like we had before, but this time they're paused in prophase 1 when we have these follicles kind of surround them. So we have these cells sort of surround them and they shut down the division process at prophase 1. This will remain paused until puberty. So, puberty. So, when menstruation occurs, there's going to be a hormone release called FSH, which is follicle stimulating hormone. Remember I said that follicles were the things that were surrounding these paused primary oocytes. And when FSH comes in, it's going to stimulate those follicles to remove and unpause the primary oocytes. When the primary oocytes are unpaused, they go through the first round of meiosis, creating 2 haploid daughter cells. But when cytokinesis occurs, one gets a lion's share of the cytoplasm and organelles. Why? Okay, so the reason why one gets it is because that one is going to become the egg, or it's more likely to become the egg. Basically, when the female's body is producing eggs, it's narrowing down that some 40,000 that started with into a select few that will have the health, resources, and genetic potential to become children. If they did the same thing that spermatogenesis did and they created 4 small precursor egg cells, those egg cells would not have the nutrients or the genetic stability or diversity to be able to produce healthy offspring. So here we're basically playing Hunger Games with the egg cells and making sure that the egg cells that do come out are going to be the very best that the body has to offer. Does this mean that mistakes cannot be made? No, it means mistakes can still be made in the development of the egg, but it's less likely and it's more likely that they will develop properly. Okay, so one gets the lion's share, the other doesn't. The one that gets it becomes a secondary oocyte and continues on. The one that does not becomes a polar body and it dissolves and dies. Dun, dun, dun. And then, of course, the secondary oocyte, which got the lion's share, is like, yeah, and then it's paused. And it's like, no, okay. And then we move on to the next step. Okay, so the secondary oocyte that's paused during, we have ovulation. So that's going to be when the female body is getting ready. It's like, all right, time to make some kids. Right, so we have the paused secondary oocyte is released from the ovary during ovulation, and it enters into the fallopian tube or the oviduct. Okay, so we said that it was surrounded by follicle cells. So more follicle cells are going to surround the oocyte, and they're going to form something called the corona radiata, which is essentially just this nutrient-rich organ that's going to help to furnish that oocyte with all the resources it needs to be able to go ahead and undergo another phase of division. If the oocyte is fertilized by sperm, if it's fertilized by sperm, it will form another polar body and an actual precursor to the egg, and the egg will form into an ovum before it fuses its nucleus. So you're like, wait, hang on, slow down. Let's go back. Okay, so we have the follicle cells surround the secondary oocyte. They start nourishing it. If the sperm comes along, that's when we're going to actually continue our meiosis and finish it and get an egg cell, essentially. If the sperm doesn't, it's going to just jettison the secondary oocyte, and the corona radiata is going to die off. And we can kind of see that here. Okay, so here we see these are the primary follicles, developing follicles. We have our oocyte here, our secondary follicle. Okay, mature follicle with the secondary oocyte, it's waiting, 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 waiting. Will it be fertilized? It releases the ovulated ovum. Will it be fertilized? If so, then this stuff will stick around. If not, then it'll degenerate into a corpus luteum and die off, and the ovulated ovum will be passed. This is what the final egg cell product looks like. Here we have our haploid nucleus, our centrioles for help and division, our cytoplasm or yolk, which contains all of our yummy, 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 yummy lipids, and we have our first polar cell, which is hanging around. It hasn't completely been dissolved and created yet. And here we have all of our follicle cells still surrounding the egg. The egg is a lot bigger than the sperm, if you notice that. Oh, and cortical granules. These will become important in just a moment to prevent polyspermy. Let's continue. All right, so you need to know how to compare and contrast these. So know this information. Notice that both of them are very similar, but in spermatogenesis, we do involve the germline epithelium, so we use that basement membrane of those testes to make the germline. We have much smaller gametes, and all of them are going to be active, and it's a continuous, uninterrupted process that begins at puberty. Here, we have a lot more stops. We have far fewer actual gametes being made. And we see that the basal germline epithelium is not actually involved in gamete production. Okay, here we have our highly, I'm just going to say, overtly sexualized chart here, which should be a lot better. You would think people would move past this, but anyway, it's, you can see the product is one ovum or precursor to the egg with 3 polar bodies, and we have 4 equal sperm. Okay, so and then we have, so there's, this is a quick little aside, when we talk about fertilization, where the egg and the sperm actually come together, which is what fertilization means. This can happen outside the body or inside. If it happens outside, it usually happens in an aquatic environment where fish sort of spurt their sperm out and their egg out into a big cloud, and then the sperm and the egg combine. This is far less likely to have there's not going to be a high success rate. Very few of those are actually going to fertilize eggs. And so there's going to be a low success rate, but it's very little metabolic cost on the cost of both parents, and very little parenting cost, because they're just like, okay, cool, my eggs are fertilized, bye. Which is very different from animals like us, which do internal fertilization, which it means that we have a higher chance of reproductive success when we do reproduce. But it also means that we have to care for and spend a lot more metabolic resources on the developing children. So take that as you will. So fertilization, male and female sex cells come together, sperm and egg. Here we go. Let's go ahead and take a look at these 3 stages: capacitation, archosome reaction, and the cortical reaction. So capacitation, if you think about it, once the egg gets inside of the vagina, once the sperm gets inside the vagina, it's going to come in contact with a lot of really acidic fluids. It's also going to come in contact with some of the metabolic enzymes that the female body is going to release, as well as some of those lymphocytes that are going to be like white blood cells almost, that are going to start attacking any sperm that are malformed. Because remember, the sperm that are coming into the female's body are, seen as being invaders. So her immune system is going to start activating, right? Okay, so when they encounter the super acidic environment, it's going to break down the sperm's cholesterol coat, so that archosome cap is going to become activated, and that cap is basically what the sperm will use to fuse with the egg. So the breakdown of the coat actually allows the sperm to have greater mobility, which we can see here. The cholesterol coat keeps the sperm protected once it's first ejaculated in, but then after it interacts with these chemicals, it is freed up so it can go ahead and start swimming for its life. The archosome reaction. So eventually a sperm will reach the egg or not, but here the sperm, let's go ahead and say it does, it reaches the egg. It pushes through the follicles of that corona radiata, which is all of the follicle cells that are surrounding the ovum, which is the not quite finished egg. It's going to bind to the zona pellicula, which is what we call the jelly coat or the jam jacket, using an archosome vesicle, which basically just means that activated archosome cap. So basically, the sperm is going to go down into this jelly coat surrounding the egg after the corona radiata. It's going to press through, digest that, and it's going to bind onto a receptor on the egg's membrane and shoot its nucleus inside. Okay, so the sperm pushes through the digestive coat, binds to receptor proteins, and then the sperm and egg membranes fuse, releasing the sperm nucleus and centrioles into the egg so that they can go ahead and combine with the egg's nucleus and centrioles and start the development of the zygote. To stop any other sperm from fertilizing, because we don't want multiple sperm fertilizing the same egg, that would create too much genetic information in an inviable zygote. The cortical granules, which are those little dots that I showed you earlier that were floating around within the egg's yolk, those are going to release enzymes into the zona pellicuda, the jelly coat, that are going to both harden the jelly coat and destroy the receptor proteins that would allow the sperm to dock with the egg, making it so that no more sperm can come in and bind to the egg. Okay, so here we go. We can see, there we go, sperm binding protein is being destroyed by the cortical granules, and the sperm can no longer enter the egg. Okay, all right, so then we have the formation of the blastocyst. So a bunch of calcium, you remember calcium from 11.2, is released to help the egg to complete meiosis too. So remember, the egg is still an ovum. It's not quite an egg yet when the sperm fertilizes it. The sperm and the egg nuclei are going to fuse, producing a diploid zygote. We're going to have the centrioles make the spindle fibers. The zygote will divide quickly via mitosis due to those spindle fibers into an undifferentiated ball of cells called the morula, which, if you want an analogy, is kind of like the apical meristem of the plants that we talked about in topic 9. The morula starts to actually differentiate and cavitate, which means that it starts to get an inside part and an outside part, and the cells will differentiate by sensing where they are along the inside or the outside, and they'll start to form into various different precursors of organs and all that sort of stuff. So the blastocyst is basically just a ball of cells with 3 sections. The inner mass is what is actually going to turn into the embryo. The outer mass just develops into the placenta, which is the organ that helps to feed the embryo. And the blastocele is going to be the cavity on the inside that's going to separate those layers to allow for the cells to be able to detect where they are and know what to develop into, and also so that it can provide, almost like the cytoplasm of a cell, a substance that they can pass enzymes and other lipids and stuff through. So it's like a transport medium, and also a medium for helping the cells to determine where they are and what they should become. Here we can see the zygote, morula, and blastocyst. The blastocyst in this case is cavitated. We can see it has the internal fluid-filled cavity, the outer layer, and the inner layer. Okay, so the blastocyst, the next big step is for it to implant into the wall of the uterus. The reason for this is so that it can go ahead and start gaining the nutrients from the mother's body, because up until this point, it has been relying upon the energy that's in the yolk of the egg to power all these reactions. So now it's going to need a greater and more long-term energy source. So it's going to embed itself here in this side of the wall right here in the endometrium of the uterus. Okay, and so here, the blastocyst breaks through the jelly coat that's surrounding it. After it starts to do all that division stuff, it releases these digestive enzymes into the endometrial lining in the uterus, and the hormones, the autocrine hormones, are going to allow it to implant itself into the uterine wall. So here we see the zygote, morula, and then the blastocyst implants itself into the wall. Okay, if you are implanted into the wall, you are going to be provided with oxygen and nutrients from the mother's body, and you will be allowed to live long term. If you can't implant in the wall, then you'll run out of yolk eventually and you'll die. Okay, all right, so here we go. So human chorionic gonadotropin, achoo, bless you. HCG is going to be secreted by the implanted blastocyst to stop the degradation of the corpus luteum. Remember, the corpus luteum is that piece that was going to help the ovulated ovum or degenerate if the ovum was not fertilized. So now that we're fertilized, the corpus luteum is going to stick around due to HCG, and it's going to produce 2 hormones called oestrogen and progesterone. Yes, oestrogen is different than estrogen. Remember that. Estrogen is just a hormone that is going to bind to stuff and stimulate a response. So estrogen is actually going to inhibit FSH, which is the follicle-stimulating hormone that paused meiosis, if you remember, and it's going to inhibit LH, which stimulates ovulation. So essentially what's going to happen is that it's going to stop other eggs from being developed in that cycle, so you're not going to continue through that whole ovulation period again, and it's going to just make sure that you're not going through like a period while you're pregnant, because you don't want to eject the developing child. Okay, and then progesterone is going to be the other hormone, and it's going to keep that endometrium from shedding itself, right? So basically, it's going to stop that implanted blastocyst from being destroyed and the shedding of the uterine lining. It keeps the endometrium intact, and it thickens the cervix so that we probably can't have another fertilization event. It's going to stop more sperm from being able to easily get up there. So basically, estrogen and progesterone are to protect the blastocyst and keep any sperm from coming back in. Okay, placental development. So remember that there's going to be that outer portion of the blastocyst that develops into the placenta. Those HCG levels are going to help keep that corpus luteum alive for 8 to 10 weeks while the placenta is developing in the blastocyst. But once the placenta develops, the corpus luteum is no longer needed. It is going to take over progesterone secretions. The corpus luteum dies and HCG levels drop. So here we can see we have HCG is being produced by the embryo. It'll maintain the corpus luteum, and which is going to do the progesterone, maintain the endometrium, and thicken the cervix. The placenta is going to allow materials like blood and nutrients and oxygen, a lot of stuff to be shared between the mother and fetus, and it's going to help to maintain the pregnancy. If the placenta is damaged or the hormones the placenta released are damaged or stopped, then the pregnancy may actually not develop fully onto term and might be terminated early. The placenta is essentially a fluid-filled sac with a bunch of veins from the embryo and from the mother going into the same space. However, blood is never directly transferred. It always goes through various different barriers to make sure that any sicknesses or immune responses from the mother are not going to super harm the baby. The umbilical cord is going to connect the baby to the placenta and to the mother. So the fetal vein is going to be where all the waste are ejected from the baby. The fetal artery is going to be collecting all the good stuff from mom's blood and taking it into the baby. The intravenous space is like the safe space where the bloods can sort of get close to each other and exchange resources. And then we have, of course, the maternal vein and the artery. The maternal vein will take any of the wastes from the mother, back and reoxygenate everything, and the maternal artery will take all the nutrients, and it's just the opposite of the fetus. So same thing. Don't worry about it. Chorionic villi are going to be all the capillaries that are extending into the intravenous space in order to take up those nutrients when the bloods are passing materials back and forth to each other. Sorry, my brain is starting to shut down a little bit as we get along. I don't want this to be too long, so I'm trying to make it quick. So the placenta works because maternal blood is pumped from the arterioles into the intravenous space. Those little chorionic villi, which are those little capillary blood vessels, are going to provide the surface area and the porosity to be able to take up all the good stuff from the maternal blood and expel fetal waste into the maternal vein. So basically, the fetus gets the good stuff and the mom gets the waste. This is what's exchanged. So we can see carbon dioxide is passed from the fetus to the mom, which is the waste product of the fetus's metabolism from its mitochondria. Water and urea is also passed back to the mom, as well as some of the hormones, excess hormones that the baby is helping to produce. And then the materials, the mother is providing the baby with oxygen, with food, with water, with vitamins and minerals, and with additional hormones to keep the baby safe and its growth regulated. So basically, you can imagine this is sort of like the mom feeding the baby once it's born. It's just happening inside. Okay? Those chorionic villi are going to be lined with even more microvilli, which are basically just little tentacles inside of the chorionic villi that are going to maximize that surface area. Those capillaries are the first point of contact with the maternal blood, but it's very important to note that the maternal blood and the fetal blood does not mix. Just the materials and resources from the bloods mix, but not the bloods themselves. And once again, that is to help protect both the mother and the child from an immune response. So here we go. Here's the fetus and the mom. This is the placental connection. You can imagine this portion right here is what we're looking at here, and here's the placenta. Okay, so 12 weeks, the placenta starts to secrete estrogen, which is different than oestrogen, and progesterone. Estrogen is going to start the growth of those uterine muscles that are going to be contracting to allow for us to actually have the birth of the child. And the mammary glands, which are, of course, going to help to feed the child. Progesterone is going to reduce the uterine contractions and dampen mom's immune system until the baby is ready to be born. In that case, the mom's immune system is no longer needed to be dampened. And the uterine contractions, we don't want to dampen those because that's going to allow the baby to be born. So progesterone is going to make sure, for the most part, that the baby won't be born prematurely. Of course, damage to the child or different hormone levels that are produced by the mother or whatever. There's a million things that could affect this and make it so the baby is born prematurely if before it happens all the time. So here we see our hormone levels. Remember, HCG is the first big one. It's going to help to preserve that corpus luteum, but once the corpus luteum is no longer needed, HCG levels decline, and progesterone and estrogen increase up until the point where the baby's ready to be born, and then those two bond so that the mom can actually have a kid. So in birth, we call this parturition. It's an example of positive feedback. If you're wondering what positive feedback is, it just means that one reaction makes a ton of other reactions happen, or makes that one reaction occur even more. A great example of positive feedback is when you go to Netflix and you find a great show that you love, and then Netflix suggests other great shows that you might love, and you start watching those shows, and those shows lead to more shows and more shows, and eventually you spend your life on Netflix. That's positive feedback. So we're going to amplify the reaction. So fetal growth is going to, of course, stretch the uterine walls. As your baby gets bigger, it's going to stress your muscles. When those stretch receptors detect those stretched muscles, they're going to release oxytocin. Oxytocin is actually going to contract those muscles, making the space for the baby even smaller, which means that the stretching gets worse and more oxytocin is released, and the muscles clench even more. And you can imagine eventually this is going to lead to the baby being squeezed out like a toothpaste from a tube. Sorry. Anyway, so you can see this is that cycle that I just said. I don't want to spend too much time on this. All right, and then of course breaking water happens. So the contractions will eventually break the amniotic sac around the child, and all that amniotic fluid will spill out, and that is what we call breaking water. The placenta itself will not be expelled until about 15 minutes after the baby's born. Here's a visualization of what happens as the baby is coming out of the mom. Yep, you can pause it if you'd like to take a look at that. The hormonal control, okay? So when at 9 months, the baby begins to stretch, our estrogen and remember estrogen, brief aside, estrogen is a type of hormone, as there are many different types of estrogen. So estriol, which is a special form, is going to make the uterus more sensitive to that oxytocin, which is going to clamp those muscles down, and inhibit progesterone, which remember was stopping those contractions from occurring. So when estriol comes, it's going to make those contractions more likely to occur. Oxytocin is released from the posterior pituitary gland inside your brain. Contraction begins, the fetus senses this, and is actually going to release more hormones called prostaglandins that are going to trigger more contractions, and this is going to lead to positive feedback until the baby is birthed. Here's a fun and confusing diagram for you to go through, and pause the video and take a look at how all this happens. Yay! Gestation period. Okay, so this is going to happen after, with fertilization, and it's going to end with birth. Lots of species have different gestation periods. It's basically just the amount of time it takes for your baby to cook and be ready to be born. And two main factors will contribute to the length of this: the size of the animal and its development. If it's really developed at birth and ready to just get up and walk around like a baby cow or something, then that's going to take a while. Okay, and if it's super huge like an elephant, that's also going to take a while. But if it's really, really teeny and is born like without most of its vital functions, like like a kangaroo or you know, like a possum or something, when those things are born, they need to develop a lot longer outside of the womb. So that's going to require less time in the mom, but more work once they're born. They're more dependent. So we call this altricial or precocial. Altricial animals have those underdeveloped helpless infants, and they have to help them survive up until the point where they can be released. Precocial animals give birth to well-developed offspring that can make it without a lot of help. As you can imagine, that increases with time. So humans are right here in the middle between altricial and precocial. But we can see that size is one of the big factors here, as the whale is like the biggest, and the chipmunk and rabbit over here have the smallest gestation period. And of course, our gestation period is a little bit longer than it should be based off of our size, but that's because we got to grow our brain matter. We got to grow our brains because we're smart and all that stuff. Anyway, cool. So that covers topic 11.4 in a whirlwind. Good lordy. Try out the practice questions and email me with questions. Woo.